Good morning, guys. We'll give everyone until 9:05, but if you want to put your name in the chat, that would be helpful. Get yourself a cup of coffee, a little breakfast snack. We'll start in a few minutes. Okay, good morning, everybody. Good morning. Happy Friday. I feel like this has been one of those weeks where it's felt like Friday, like every single day. So I'm happy we finally made it. Okay, so what I'm going to do is I'll show you Canvas, kind of what we got going on for what amounts to the end of the semester. And then I received some questions ahead of time, so I'll go through those questions and then we'll do our activity. So again, for those of you just joining us, please make sure you put your name in the chat. So when I download it, I have a nice written record of who was here and who was here on time, all that good stuff. Okay, so again, good morning, put your name in the chat. I'm gonna share my screen here for a second. Let me pull this down. Get this together. Just, you know, getting ourselves together this morning. Okay, so we're here in Module 6 in our senses. Last week we talked about vision. We spent a fair amount of time going through vision with a focus on how we perceive color and how we see color and what goes wrong in people who have a color deficiency or are colorblind. And so today we're going to focus on our ears, and we're not going to spend a lot of class time talking about our other special senses, but you did hopefully read about them when you did your guided reading questions. So as a reminder, the module is ending on Sunday, so you're going to have that homework due. You should have done the discussion post, and you're going to have a comment on a peers post by Sunday as well. And then next Monday will be our last quiz. So quiz on Module 6. Again, it's due at 6 p.m. So please make sure you set like multiple phone alarm sort of situations. Tell your roommate to remind you, all that good stuff. Okay, so last quiz. And then next week, we're doing the endocrine system, and it is going to be brief. We're going to focus on just two parts of the endocrine system. So we'll talk about our pancreas, and then we'll also talk about the adrenal gland and what the adrenal gland does. So just two systems there. And for this chapter, we're not going to have a quiz. We're not going to have a discussion post. You're just going to have one guided reading question page. Well, it's a few pages, but one guided reading question assignment, and then a homework. And it's a slightly shorter homework than usual as well. So that will take us to next week, or the week after. So November 9th and November 11th, we're having that in-class final exam. And again, it's going to be cumulative. It is 12 short answer questions. So your final exam is just 12 short answer questions. They are spread pretty well dispersed. There are going to be at least 2 questions on this endocrine system unit just because you don't have a quiz on it. So it's going to be slightly weighted towards module 7, but the rest of them will be pretty evenly distributed in modules 1 through 6. So it'll be 12 short answer questions, no multiple choice. So what that means for your studying, you don't have to study the multiple choice because it's not going to be on the test. And so there won't be questions about labeling muscles. There will not be labeling bones. You won't have to label anything. Instead, I want you to think about some of the big themes that we have talked about and covered, that you've seen in your homeworks, that you've seen in the discussion posts that we focus on in class. Those sort of like big topics. That's going to be more of the focus for your final. And then also as a reminder, you have the final project you're working on. Your abstract is due. And then again, you'll be making a poster and doing a very short presentation on your topic in the time period that's reserved for our class to take a final exam. So during exam week, we will instead have a virtual poster session on Zoom. So that's where we're headed. I am going to switch gears here. Let me pause. Are there any questions about what is coming up, what's on the horizon, what we got going on? If you do, you can unmute yourself and ask, or you can put them in the chat. I'll give everyone a second. Okay, while you think about any questions, I'm going to switch over to the PowerPoint I made about the questions I received. Okay, so, sorry, I've got like 5,000 windows open. Okay, make sure I have the chat up just in case. Okay, so I had received a few questions I want to go over, mainly about how does hearing work, how does balance work, what do our ears do? So let's think about our ears for a second. When we're thinking about our eyes, right, we were focused on light waves coming through, hitting those photoreceptors in our retina and being processed by our brain. So for our ears, we're also talking about waves, but we're talking about a sound wave. What was the attendance quiz? I haven't gotten there yet, patience. I had received a few emails of a few people running late this morning, so I was going to do the attendance quiz after I do this PowerPoint. Yeah, I know, it's, you know, yeah. I had received a few panicked, like, it's a rough Friday email, so we're just going to do the attendance quiz after I talk for a little while. I cannot see your screen. Can anyone else see my screen? Does anyone see a PowerPoint here? Can someone tell me yes or no? Yes.
Yeah, I can see it.
You can see it? Trinity, I don't know what's going on.
I don't know what's going on either, because it just says Courtney Carroll has started screen sharing and it's like not showing anything.
Not showing anything? Okay, well, this PowerPoint is on the class agenda, so you can like open it on your own screen and kind of follow along while I explain stuff. That's kind of the best I got, unfortunately. Okay, so let's talk about sound waves for a second. Now, I know physics is a scary thing for a lot of people. It's a little scary for me too, but we're going to get through it together. So sound is a wave that propagates through space. And it comes first to our auricles, or our outer ear, where it's going to be funneled in and it's going to pass through that auditory canal, and it will hit our tympanic membrane, which is the science word for our eardrum, right? And it's going to send that vibrating with that same wave function, that vibration. The ossicles in our middle ear are then going to move and amplify that wave, make it stronger, and it's then going to hit the oval window in our inner ear. And the difference here is that our inner ear is full of fluid. And so what that wave will do will be the same thing if you dropped a pebble into a pond. You're going to send out little ripples. The same thing is going to happen in your ear. We're going to apply a force and send a wave function through that fluid. And we're looking at the area called the spiral organ of Corti in that cochlea in our inner ear. Remember, your inner ear is in your temporal bone. Can everyone touch your temporal bone for me, please? Everyone touch your temporal bone. As a reminder, the final is cumulative, so let's make sure we understand the basics of bone stuff, right? We have temporal bone, frontal bone, parietal, occipital. Vision is processed all the way in the back in the occipital, but sound is processed in the temporal lobe in our temporal bone right here, okay? So we're in the spiral organ of Corti. We're going to talk about a few different structures. The first one we want to talk about are the receptors here. So these are mechanoreceptors. If you remember, for vision, we were talking about photoreceptors. For taste and smell, those are chemoreceptors. Here with hearing, they're mechanoreceptors. And they're little hair cells, and they're on a structure called the basilar membrane. And as the wave function moves through the fluid of our inner ear, it's going to move something called the tectoral membrane, and that will then push the hair cells. And when the hair cells get pushed over, that's what sends that action potential. So if I tell you that the hair cells are firing action potentials, we want to think back, let's think back to the beginning where we talked about our 4 kinds of tissue. We have 4 different types of tissue. We've got epithelial, connective, nervous, and muscle, right? Firing an action potential and having that sort of electrical signaling, that implies that these hair cells are nervous tissue. So we want to try and put all this in context, right? So they're going to fire an action potential. We learned about action potentials in the last module. You want to keep in mind, you still need to know the steps of an action potential, right? Sodium, potassium, calcium, what all of those ions are doing. So that impulse will then be passed through our cochlear nerve, which will then join with our vestibular nerve, and it will eventually go to our auditory cortex in our temporal lobe. So let's see what that looks like. So this first picture, you can see the little hair cells on the basilar membrane, and as the fluid moves, it's going to move this tectoral membrane, and it's going to bend these hair cells, sending an action potential down our cochlear nerve. Okay, so again, vibrations from the sound waves will then move the membrane, the hair cells will bend, and then we'll have that action potential, and it will be processed in this pathway of first going to our brainstem, through the auditory pathway, eventually to the auditory canal or cortex. Okay, let's look at it one more time. So again, we have these little hair cells on our basilar membrane, we have our tectoral membrane. What I want you to see here is what I mean when I say mechanoreceptors. Mechanoreceptors are pulled open. And so the hair cells are actually connected to each other by these thin filaments, these thin protein fibers. And when the hair cells are nice and parallel to each other, the channels are closed because there's no force being applied. But when the hair cells start to bend, that filament is going to kind of pull open and force open the channel, and it's a cation channel. So cation, remember again, cation means positively charged. Positive charges will go into this hair cell, depolarizing it, starting off an action potential. Okay. Now, a lot of people asked about what do you mean with how we encode pitch and how we encode volume. So this, again, we have to think about what the wave function of hearing means. So we have a wave function, and there are different properties for that wave, as someone is drawing on the screen. Thank you. You know, Zoom is fun. So the wave function, if the waves are close together, if the wavelength is short and those waves are close together, that's a high-pitched noise. So it's a really squeaky high-pitched noise. And the high-pitched noise is different from a low-pitched noise. Low-pitch, again, wavelengths are further apart. We've had a long wavelength, low-pitched noise. They're going to bend different hair cells. So the high-pitched noises are going to bend the short, cute little hair cells right next to the oval window, and the low-pitched noises are going to bend the long hair cells far away. We encode volume, which is the other component of our wave. So we have wavelength, which is going to tell us about frequency, which is the pitch. And then we also have volume, right, which is our amplitude of the wavelength. And that is encoded based on the frequency of action potential. So if it's just a whisper, it's super, super, super quiet, it's just gonna bend the hair cell like once, right? And that's gonna be it. If it's really, really loud, that hair cell is gonna bend over and over and over again and fire multiple action potentials down that hair cell. Okay, so that's how we encode pitch versus volume. Let's look at that in a picture form. So again, high pitch noises are these little short hair cells. Long noises, low pitch noises are these long hair cells far away from the oval window. Okay. The other questions I got were about equilibrium. So a pet peeve that I have, we all have pet peeves. One of my pet peeves is when I ask a question about equilibrium and we're talking about our ears and I get responses about the balance between reactants and products in a chemical reaction. Technically, a definition of equilibrium is the balance of reactants and products in a chemical equation. However, that is not what I'm talking about in this class. And so if you give me answers and short answer questions, I usually give some partial credit. However, if the question is about equilibrium and you tell me about a chemical reaction, that gets no partial credit because it just makes me very upset on the inside. So here we're talking about equilibrium, meaning balance, okay, in our inner ears. So for static equilibrium, we're in an area called the vestibule. Static equilibrium is your head position in the up-down direction. The receptors are called maculae. And what we're detecting here are still mechanoreceptors, but rather than detecting the movement of fluid, we're detecting the movement of salt crystals. And these salts are called otoliths. So when these otoliths move, they will then move the hair cells, and then that will fire onto our vestibular nerve, which again joins with our cochlear nerve, cranial nerve 8. So again, you bend your head down, you move those otoliths, you bend your hair cells. Your body is aware now that you're moving your head. Dynamic equilibrium or rotational equilibrium is slightly more complicated. So we have these structures that are called semicircular canals, and these are located one in each plane, in the X plane, the Y plane, and the Z plane. And when you rotate or you move in a certain direction, it will send the fluid moving in that canal. And then that fluid will end up in this ampulla area, this ampulla structure, which has a specific region in it called the crista ampullaris. And inside the crista ampullaris are cupula, which are the name we give for the hair cells for dynamic equilibrium. So again, here are our cupula in our crista ampullaris region in the ampulla. And we're here talking about the movement of fluid. So we're going to move fluid when you spin around in a tight circle, and it's going to bend the hair cells, and your body will then interpret that as rotational movement. Okay. I am going to stop sharing this. Do I have any questions about that? I know I went through that kind of fast. Okay, again, we're in our inner ear, talking about hearing, and we're talking about equilibrium, and I mean balance. I don't want to see any responses about chemistry, or I'm going to lose it. I just will. It'll be very bad. It'll be bad for all parties, okay? None of that. Okay, so now let's get to our attendance quiz. I think everyone has joined us. So if we're in our, let me just move my screen down so I can see my little bar here. Okay, we're in our class 12 agenda. If you could click on your class 12 attendance quiz. And if you could tell me, we just went over it. What are the two kinds of equilibrium? What are the two kinds of equilibrium that we have? What are the two kinds of equilibrium? Give everyone a minute for that. Okay, so the two kinds of equilibrium again are static and dynamic. Static, head position in the up, down, right, or with respect to gravity, and dynamic is rotational. Okay, great. So we're first going to start with a case study. So this is about a person who is having some issues with fluid in the ear, or he suspects fluid in the ear, and he has a few different issues with that, and it's got 5 parts. It's got part 1 here, part 2 is got some test results, right? And then there's a label and ear picture. Part 3, and then if my internet can load, there's a label, the inner ear structure here, part 4, and then we end up with part 5. Okay, So what we're going to do is I'm going to break you guys into 5 groups, and each group is going to work on one of the different parts, and you guys are going to put your answer. If you open up this Google Doc, it brings you to a place to put your answers. So group 1 will work on part 1, group 2 will work on part 2, group 3 will work on part 3, etc. And you guys will talk about it, like with words from your mouth, and then put your answer here. The ones you don't have to do are the labeling ones. There's really no way for us to do labeling really well. However, I think it's helpful for the Module 6 quiz. Make sure you can label the ear, for example, label the inner ear. So make sure you are able to do that. And you guys will work on this for a little while, and then we will come back together. So I'm going to put you guys into those 5 breakout rooms. Okay, work on this for at least 20 minutes on your part. Talk about it, and then come up with an answer, and then we'll go through it. Okay. Open up those rooms, get to that. If you have any questions, let me know. Elise, you okay? There we go. Tyler, you doing okay? Okay. There we go. Okay, you guys are doing part 1. Any questions? Any issues? You're good. Okay, I'll leave you to it. Okay, you guys are doing part 2. Any questions at all? Any issues? Okay, I'm gonna assume everything is okay. Okay, you guys are doing part 3. Any issues? Any questions? No. Okay, I'll leave you guys to it. You can put any questions you have in the chat.
Okay.
Okay, you guys are doing part 4. Any questions?
No, ma'am.
I'll leave you to it. Okay, you guys are doing part 5. Any questions?
No.
No, ma'am.
Okay, I'll leave you to it. If you have questions, you can put them in the chat.
Thank you.
Welcome back. So again, we're talking about our ears. We're talking about what can go wrong with our ears. So let's jump over to the Google Doc and see what you guys came up with for that. Okay. So the first part, we're introduced to Julius and some of the problems that he's been having. And so the first part, what problems is Julius experiencing? Headaches, pressure in the ear, hot and nauseous, balance is disturbed, right? Exactly. Are any of these problems related? Yes, it's related because normally when your balance is disturbed, you can get nausea and headache as a symptom. Yeah, so all these things are related to each other. And we know this because we know that in our inner ear, we have both hearing and balance. And so all of those things kind of work together. What are some potential causes of these problems? Drug use, drinking while antibiotics, fatigue. Yes. So the doctor he sees is especially big on that drug use issue, but it could be drugs, it could be drinking with antibiotics, it could be stress, it could also be diet. And so question 4, how could his eating and drinking habits cause or perpetuate his current symptoms? He is not eating the right foods. Yeah, so this becomes a chronic problem. And you guys all know this. We're entering the end of the semester here. When you're in times of stress, oftentimes you're also not eating right, you're not sleeping right, you're not exercising, you're not taking care of your body, you're not drinking enough water. And so all of these problems can kind of compound themselves, especially high salt foods can cause that high blood pressure, same with dehydration, all of that. What test would you like to run? A CAT scan and a blood test. They're going with the drinking problem. I think it's alcohol. So yes, odds are they're just going to take a bunch of blood. This is what doctors do. You go to the doctor for anything and they're going to just remove all of the blood from your body. So that's what they did. So part 2, what new signs and symptoms is Julius experiencing? Dizziness and headache, and the pounding in his ears is worse. Yes, exactly. So that has gotten worse. And then it shows a table with some of his test results, which ones are not within the normal range. Yeah, so he has high blood pressure and high white blood cell count. He also has a slightly elevated sodium level and a slightly elevated heart rate. What might explain these abnormalities? It could be the antibiotic he's taking. It could be the antibiotic. It could also be that stress, the poor diet. But the thing we want to focus on is the white blood cell count. So whenever you see high white blood cell count, that means your body is battling an infection of some kind. It doesn't mean it can't tell you like a specific location for the infection or what type of infection it is, but high white blood cells means that your immune system is battling something. And so that tells us that there is some sort of infection going on. If you were an ER physician, what additional tests would you run? So you could run a whole bunch of tests, again, more that blood work, that CT scan. You could also run some specific hearing or balance tests to see what was going on. And then in question 6, yeah, we're labeling the diagram. So you guys want to make sure that you are able to do that, to label the parts of an ear. Great. So part 3, what could possibly go wrong? So again, part 1 is another diagram situation. Question 1 for part 3. Make sure you can do that as well. Question 2, where in the cochlea are the hair cells, the sensory cells? Yes. So they're in the spiral organ of Corti, specifically on the basilar membrane. Exactly right. Perfect. Question 3, what is the function of the stria vascularis, the cells that line the scala media or the cochlear duct? So we didn't go into this in particular, but for this, just know that these are the areas that secrete and regulate and recycle the endolymph area. So the endolymph, again, is that fluid that's moving throughout. So the endolymphatic sac is a blind ending structure connected to the cochlea and balance organs via the endolymphatic duct. One function of the sac is to secrete endolymph back into your circulatory system. If Julia's problem is produced by an increase in the pressure of that endolymph, what factors could contribute to an increase in volume in this fluid? So it could be, yes, that there's too much endolymph being produced and there might not be enough drainage of that endolymph, and that could be an issue. And so, yeah, it could be a lot of the problems that we're working on could come from that inner ear pressure. Again, your inner ear is responsible for both hearing and balance, and when those get out of whack, you can very easily get a headache and issues with blood pressure and all of that. Okay, let's move to part 4. Remember that the level of potassium in the endolymph is higher than inside the hair cell, and so we're going to use the word bank here. So Bending the stereocilia opens the channel and potassium ions enter the hair cell. The membrane will depolarize. The voltage-gating calcium channels open and calcium will enter the hair cell. The calcium ions cause the duct vesicles to fuse with the membrane. The neurotransmitter binds to receptors in the postsynaptic membrane. Depolarization of the membrane of the sensory neuron produces action potentials, and axons travel in the vestibular cochlear nerve to the brain. Exactly. So this is a slightly different action potential than the one we talked about, the classic action potential. And that's because of potassium. And what they point out here is that the concentration of potassium in the inner ear is high outside the cell, low inside the cell. So rather than being sodium that enters and drives that depolarization, it's potassium. And so you just need to be able to modify in your head what you think of the steps of an action potential based on those concentrations. Okay. Part 2, the ion composition of the perilymph is similar to the interstitial fluid, high sodium and low potassium. So Dr. Luther stated that Julius's fall ruptured his vestibular membrane and the membrane between the scala vestibuli and the scala medium. What would the effect of such a rupture on the concentration of the potassium in the endolymph in the scala media when it mixes with the perilymph? So it could either mix with the sodium that would make it equal concentration, or it could increase the level of calcium. So these two solutions are going to mix, and the end result is that there will be lower potassium than there needs to be. Because if you think about chemistry, right, we're mixing these two solutions, diffusion will happen. And so we'll go from an area of high potassium to, it'll even out to be like a medium or like lower amount of potassium. What would be the effect of this mixing of endolymph and perilymph on the amount? Oh, I just said that. So yeah, so we're going to lose that concentration gradient. Yep, and we're not going to be able to get that depolarization to happen as quickly. Okay, if the number of open voltage-gated calcium channels depend on the amount of membrane depolarization, what would be the effect? So again, if we're losing potassium, we're losing that driving force here for the depolarization just in the inner ear here, then we're not going to have as many open calcium channels. We're not going to have as much signal. So what I want you guys to think about is to think through effects that would happen by changing one thing in a process, right? So for example, we talked about the classic steps of an action potential, sodium in, potassium out. Now remember, here in the inner ear, it's backwards because of the fluid, so don't mix those two up. I know it's similar, but in a normal action potential, sodium comes in, potassium goes out based on those concentration gradients. And so you need to be able to think what would happen if you alter one step in that process, what would happen to the rest of the steps. Okay, part 5. The audiogram indicates that Julius has problems hearing certain frequency. What type of damage may have taken place? Inflammation. Yeah, so he has that inflammation, and it is unlikely that there's damage to the outer ear or the middle ear. And again, we know about frequency coding in the hair cells, so we can predict based on what the frequency issues are, which hair cells may have been damaged. Would this damage necessarily have taken place from both ears? Yes, because the duct, yes, so it's affecting, it is connected to each other, and so we would predict that there would be damage in both ears. If you were a doctor, what changes would you recommend? Living with your parents, eat healthier, and start, yes. You want to maintain a low sodium diet, low blood pressure. So again, if we're talking about fluid buildup, we got to keep the blood pressure low. We got to keep water at a key level, and we got to watch our sodium. Okay. So for the class 12 assignment, if you guys could just screenshot whatever section you worked on and submit that. So if you worked on part 5, screenshot part 5. If you worked on part 4, screenshot part 4, etc. The other thing we were going to do, and we don't really have time for it, and it would be hard to do virtually, but I put the videos up here. We're gonna watch a video about the history of ASL, American Sign Language. We're not gonna do that, but if you're interested in sign language, there is a video here about the basic signs, and it's right here. She has a really good Instagram as well, if you are interested in that. Sign language is something that I think is useful. Learning a language in general is useful, but sign language in particular, if you go into a medical field, you might encounter a patient who is deaf, and knowing just a little bit of sign and being able to communicate with them a little bit is helpful. The other area where it's helpful to know sign language is if you work with young kids or kids on the autism spectrum, because for a lot of nonverbal kids and people, sign language is a way that they communicate. And if you work with young kids as well, some parents and educators will teach their kids sign even before they can speak or if they have issues with speech. And so knowing a few basic signs, I think is really a helpful thing. So I have that here. And then the last website is called Signing Savvy. And this is an ASL dictionary that I think is really great. It's fun. I took sign language in college. It's now been about almost 10 years, but I think it's a fun online dictionary. You can see the sign of the day is apple, so you can type in whatever phrase you want and see how to find it. They have a special one for the holidays, right? So that's just like a fun thing you can do. Okay, so make sure you submit your class 12 assignment as that screenshot. And if you guys have any questions, I'll stick around. Otherwise, you are good to go. And I'll see you next week. We have our last lecture. How crazy is that?
Is the screenshot the only thing we have to submit?
Yeah, that's it. 'Cause we didn't get to the videos. So yeah.
Okay.
Just the screenshot. I apologize for the lawn care going on in the background.
Hey, I have a question, but it's not about this class. For our abstract that is due on Sunday, I was wondering if there was a specific person you wanted us to like discuss in, because I know in typical abstracts we don't use we, I, or you.
I don't care.
Okay, just making sure. Thank you.
It'll be in sentences. I care
Okay.
about that, but I don't care about tense or citation or
Any of that? Okay, just making sure.
yeah, you're
Thank you.
Brendan, what's up?
I was going to ask, what was the file types that we could upload? Because I know you said that your computer would only take certain types. I couldn't remember
It just, so I'm on my home computer right now, so I can take any of them because it's a Mac. But when I grade at school, I can't do .heic files
Gotcha.
because they're like only Macs.
Okay. Cool.
Okay. You're welcome. Enjoy your weekend.